My name is Hilary Godwin, and I'm a professor in the UCLA Fielding School of Public Health and the Institute for Environment and Sustainability at UCLA. I've been with the UC CEIN since it began in 2008, and I have oversight responsibility for our education and outreach activities within the center, and also do work on high throughput screening in bacteria. The basic research that I'm doing within the UCCEIN is focused on looking at how nanoparticles impact bacteria. Bacteria really form the foundation of ecosystems. It's incredibly important, not just for human health, whether or not nanoparticles are interacting with the bacteria that, for instance, help us digest our food, but also whether nanoparticles out in the environment are impacting organisms that form the basis for many different living systems. So within the research that we do, we've looked at how a series of different types of nanoparticles impact the growth of bacteria, and also done detailed studies to try and understand why they have those impacts on bacteria. Those particles that we're looking at include particles that are intentionally used as antimicrobial agents. So silver nanoparticles and copper nanoparticles that are used commercially for applications in which people are trying to kill bacteria. And we want to understand both how to design materials that work effectively for that application, but also don't unintentionally kill bacteria that we need, for instance, to keep us healthy, or adversely impact other organisms at the same time. One new discovery that we have is looking at the impacts of a variety of different metal oxide nanoparticles on bacteria. Metal oxides are used extensively commercially and appear in a variety of different products industrially, but also in some personal care products such as sunscreens. And we looked at a series of 24 metal oxide nanoparticles that had been looked at previously by other researchers in our center to see. Other researchers had looked to see how they impacted, for instance, human cells, and we wanted to see whether or not the same patterns held true for bacteria. And the interesting result that we found was that the patterns that we saw in terms of which particles tended to show toxicity in the human cells were paralleled in the bacterial cells. So you may be thinking, well, of course, they're both cells, so they're both being affected the same way. But actually, one of the very important premises of the field of toxicology is that we don't expect necessarily to see the same patterns in species that are very different from each other, that there could be different mechanisms and hence different patterns of toxicity. So it was actually interesting for us that in this case we saw virtually identical patterns, and that tells us that when using those particles, that we could actually use species as simple as bacteria to screen for safety of those particles and probably get good predictions about what would happen in a variety of other organisms as well. My parents are really responsible for my early and continued interest in science. I have four parents. My biological parents both remarried when I was fairly young, and so I have a stepmother and a stepfather in addition to a mother and a father, and I'm close with all of them. And all of them are biologists, and so from a very young age, I really learned directly from them their love of science and love of exploration. They involved both my sister and I in their fieldwork activities when we were little kids. And so I really have always had a very hands-on appreciation for science and discovery, and also for the potential for exciting young people about science. And I think that helps to explain both why I've ended up doing, even though I trained in chemistry, I've done stuff that overlaps with biology, and also why I have this continued interest in sharing my enthusiasm for science with young audiences. One of the best pieces of advice that I received from, I don't think it was even really meant as advice, but that I received from my thesis advisor in graduate school was actually a comment from him that he thought that his best students were, or most successful students were the ones who were the most persistent, as opposed to the ones who were necessarily the most intelligent or the most genius-like. And that really hit home with me when I was in graduate school, because we were all trying to be the smartest person, and being the most persistent really wasn't on my radar screen. And I have to say, over the course of my career, I've definitely seen that played out in a number of circumstances, and it certainly would be my advice to young scientists, which is that sometimes it's just more important to be persistent, or as I like to say, persistently pleasant, in terms of accomplishing things, than it is to necessarily have the most brilliant idea. The ability to stick with a problem even when it's frustrating, and to push through on that problem, and to continue trying different approaches can be really important to solving scientific problems and life problems, sometimes more than that sort of streak of inspiration.